Hey.
Hey.
So, we yesterday created this beginnings of a T cell model in which we use CD3, side scatter, CD4, and CD8 to select just CD8 positive T cells.
That's right.
But we left it there. We didn't do anything about staging and setting up the progression markers or anything like that. So, I'm in the mood to set up some progression markers.
Okay,
How's
Ben.
that?
All right. Um, so this is actually the way we normally build models. We first just build the part that deals with the selection, test it on a whole bunch of files, make sure that that's working well. Then we go and we add all the other markers to it. Just ends up that if it works well with the selection, then it's actually an easier problem, believe it or not, to add all the other markers to it and have
Sure.
those work properly. So let's get started. So we'll go ahead and click gemstone here, say OK, and we'll take a look at the batch system. And this is where we left off,
Yep.
Ben. And in order to re-execute the program with that data that we were using, I'll just go ahead and double click. It's loading in the data now.
It's like a million events or so, so it's a big file.
It's a quite, especially for T cells, usually you need quite a number to do this kind of stratification work. All right, so let's put this away here, get a little bit extra room. Now we're going to add the three markers that we're going to use for staging. And those are CD28, CCR7, and CD45RA.
And you're going to add them in that order, I'm assuming.
I am, and remind me to explain why. And then once those three are selected, we'll just go ahead and get the rest of them here. So CD57, CD27, CD56, and we'll throw in a little dash of scatter.
Okay.
All right, so we start with CD28, and a good question would be, okay, why? Why would you start with CD28? And the reason is because it kind of divides the population into two halves. So we did the same thing with B cells. We started with CD10, same idea, just divide up the population into positives and negatives and have those roughly be the same. It ends up that that's really one of the best first moves you could make when doing modeling. So we'll go ahead and click our magic hat here and choose step down. And you can see, Ben, as soon as it has done this fit, we can see that there are a lot of other changes taking place in all the other markers.
Right.
And some of them are really, really interesting. We see a little increase in scatter. CD8 comes down a little bit. I think I mentioned in the previous tutorial that 8 and 3 do modulate, and you can actually see them changing a little bit here. Let's go ahead and do our next step, which would be CCR7, and you can see that it's suggesting that it's a step down.
Yep.
So we're good. So we'll go ahead and choose that. And really, as soon as it has that initial directionality data, pretty much everything else is determined at this point.
And it looks to me like if the program is suggesting a certain profile, a step down in this case, that it also knows where it wants to position those two steps, and also it looks like it knows how it wants to initially set up the line spread for them. There's a lot of information it has behind that recommendation.
Right. These little triangles, these black triangles, Ben, are a special peak finding system we have. And so when we add an expression profile like this, goes ahead and draws information from those peaks. And that's the initial position, the line spread, and then it does its fit, and after doing the fit, we can ascertain what comes first, CCR7 or CD28, as far as downregulation. So here we've done 28 and CCR7, and now we just have CD45RA. And you can see here, Ben, that the mistake that I could have made here is I could have started with CD45RA, and that would be hard because it's high, then it's low, and then it's high again.
Right.
So there'd be no, it would be hard to model that as your first step. So we typically do simple things first to get that directionality, and then we do more complicated expression profiles later. So here it's asking for 3 levels. We'll just go ahead and do this.
We talked about this in earlier movies, Bruce, but what's happening here each time you add one of these is that the program is reorganizing not just the events that are in the one plot we're analyzing, but if it moves an event from one plot, it's moved in all of the other plots. It's along the same x-axis for each event.
Right. Another way of saying that is each event here is vertically aligned with another event in other events in other expression profiles.
Right. Okay.
So when they move, they all move together. All right. So at this point, we have this nice overlay, and we can see what's happening with all the markers. What we normally would do now would be to stage it, and we'll, as we did in the B-cell tutorial, we'll go ahead and do this part automatically. So at this point, it has staged the data, and you can see that it actually ends up simplifying the look of the overlay because the labels are now just in the middle of the stages,
right,
instead of what we call our wire labeling mode. So really at this point, Ben, we're done with the template. So we'll go ahead and save that. And as usual, I'll just add a version 2 to this. And then in the batch system, we'll go ahead and just tell it that we wanted to use that template instead of the first one we did.
Right.
So that's pretty much it. At this point, we could go ahead and automatically analyze the other files, but I wanted to just show you one more thing, which I always enjoy demonstrating, and that is that we have quite a number of markers here. Why don't we look at all the dot plots associated with those markers and then see the progression in all those dot plots. So to do that, we create what we call a SPLOM. And then normally I turn on the animation just so you can see all these distributions, both with color and with motion. It's so you can see it's quite a
There's a lot of plots there.
A lot of plots, and the important thing is that all the information and all those other dot plots is being shown to us in our overlay.
Yeah, that's amazing.
And this one's a lot simpler to look at since it's really showing you the biology directly. All right, so we have our template. Let's see if it analyzes the next file correctly. And say that this data comes to us from Margaret Inakuma at DD Biosciences. And I point that out because it's really hard making really good high dimensional cytometry data. And this is just nice. And it makes the modeling a lot easier. Okay, so it finished this one. And you can see there's similarities between different donors. But the width of the stages and everything changes just because people have different percentages of these different stages.
I see, and that's really what we're seeing on that progression of the overlay plot is the stage width is relative to the percentage of events in that stage.
Exactly.
Okay.
And that varies considerably from person to person. Okay, let's try the last one. So it's going through its selection. It's automatically doing an enriching at this point. And now it goes through and it starts stratifying the data. And it's doing its final fit, and we're done. And you can nicely see, Ben, that these percentages actually can dramatically change from person to person.
Yep.
This is stage 1 is the naive stage, and you can see that in this particular donor is actually quite small relative to the other ones. So that's pretty much it. The first tutorial was in many ways the hard one because we need to make it so that the selection is very robust. But once we had that template model, it was relatively easy for us to add the staging markers to it and add modeling to the system.
That's fantastic.
All right.
Thanks, man.
Okay.